Hello everyone, so we're going to be learning about proteins and you're going to need the following: your Unit 2 book, pen, paper for some of the past paper questions and some of the other work, and then also a data book. Certainly for the first couple of questions you might want to use a data book. Okay, let's get started. So 2 questions here to get us going. Have a go at these, and when you're ready, unpause the video. Alright, so the first one is embarrassingly easy. So the elements nitrogen, oxygen, fluorine and neon. If you don't know this off the top of your head, check your data book and you should have found that they are all gases at room temperatures. They don't have single bonds between the atoms because neon is monatomic, so that doesn't fit that one. Again, neon is not diatomic because it's monatomic. The others are diatomic elements. So can they form negative ions? Well, nitrogen can, oxygen can, fluorine can, but neon again cannot. It's unreactive. Okay, so 1 is D. Next, which of the following equations represents the first ionization energy of fluorine? To get the answer for this one, we need to know our definition of the first ionization energy, which is as follows. So have a read of that there. And the only one that fits that is, well, it can't be A, can it? Even though we're removing 1 mole of electrons from a fluoride ion, we're not moving it from an atom, so F negative there, that's what we're starting off with, that's the fluoride ion. Everything's in the gas state, so that's fine. In fact, everything for all of them, A, B, C and D are in the gas state, so it does fit that. It can't be B because again we're starting with the fluoride ion, and it can't be D because we're starting with a molecule, so half a mole of fluoride molecules. So the only one that fits is C. We've got a fluorine atom in the gaseous state. We're removing an electron and it's forming a gaseous ion. So C is the answer. Really tough, this one. Pause it. When you're ready, unpause. Okay, so when I initially read this one, I went down the route of propanol plus ethanoic acid. Okay, so we've got an acid and we've got an alcohol there. We react those together, get an esterification reaction going, condensation reaction, and we're going to form propyl ethanoate. So that was my initial thoughts, and water. But then I realised that we've got NaOH is being added. Well, to make an ester, we need an acid catalyst. So H2SO4 is the one that we used in class. So it isn't going to be an esterification reaction. So then I looked and thought, well, what's the NaOH going to react with? Will it react with the propanol? Probably not, no. Is it going to react with the acid though? Well, NaOH is a base, ethanoic acid is an acid. So we've now got an acid-base reaction, a neutralization reaction happening. So what are we going to have in our beaker at the end of it? Well, we're going to have a salt going to be formed. We're going to have, which one is it? Sodium ethanoate. And we're also going to have water. And we're going to have propanol. So the propanol isn't going to react at all. So A is the correct answer. Okay, next one. Okay, a little bit easier. We've got this chemical here. What are the functional groups present? So we have 2 different functional groups. We have these 2 here, and one of them is very, very familiar, the OH. We should know that as the hydroxyl group. So now we're looking at between C and D. Do we have a carbonyl or a carboxyl group? Well, the C double bond O is known as a carbonyl group, and if we had an OH attached to the carbon there as well, as opposed to just the hydrogen, we'd be talking about a carboxyl, but we don't. We've just got the C double bond O. So we're talking about a carbonyl group and a hydroxyl group. C is the correct answer. This question here, can you define condensation reaction? So here's your answer here. 2 molecules, so that's the 3-methyl-butanol and propanone. They join together and we have lost a smaller molecule, in this case water. Okay, so there's your answer there. So on to proteins. So what we're going to cover today is the following. We're going to give uses and examples of proteins in organisms. We are going to draw and label an amino acid. We're going to define essential amino acids. state or show how amino acids join to form proteins, and then vice versa as well, how proteins can be broken apart to form amino acids. And then lastly, explain how proteins become something called denatured. So explain how proteins become denatured. So that's what we're going to cover in today's lesson. The sort of questions we can expect are as following. So something called essential amino acids here. Can you define that? We've got a long structure of a piece of protein here. Can you circle something called a peptide link in this? So we're having to look at the structure and identify different parts of it. So those are the sort of questions we can expect, and we'll go through them later on. So just a little revision quickly of condensation and hydrolysis reactions, because we're going to come across those. You may remember when we looked at esterification that you can react a carboxylic acid and you can react it with an alcohol, and if you do so, you'll remove the following atoms. You'll remove the O and the H, so the hydroxyl part of the carboxylic acid, and also the hydrogen from the hydroxyl of the alcohol. And excuse me, if you remove those, you will then form, in this case it's an ester bond or an ester link, and then also water is being removed, and that reaction is reversible, and reverse is known as hydrolysis. So a little bit of revision about condensation and hydrolysis there. And why is it condensation reaction? Well, just as we showed before, two molecules join together with the loss of a small water molecule. So we get proteins from a variety of different sources. They're essential for our well-being, for our health. We can get them from nuts, from meat, from dairy products, eggs, and so on and so on and so on. And they are essential. They're really, really important for our health. We use them for a variety, the body uses them for a variety of different tasks. So for example, enzymes, we're going to come across enzymes in a little bit, and support, so structural support, hormones as well, in DNA and RNA, and so on and so on and so on and so on. So we have some uses there of proteins. The way that the body makes proteins, the way that it makes muscle, for example, is it takes small molecules, and it takes those small molecules and it joins them together to form one big molecule. We refer to the small molecule as a monomer and the larger molecule that's formed as a polymer. Now, the type of reaction that happens when we get these monomers, they join together to form a polymer via a condensation reaction. And because we're forming it via a condensation reaction, we refer to the polymers as condensation polymers. So the monomers that we use to make proteins are known as amino acids, and the amino acid is characterized by four parts to it. We have, first of all, one of the functional groups known as an amino group, the NH2 part there. On the opposite side of the molecule, we've then got a carboxyl group. And then we've also got something there called a side chain, which is represented here by R. And of the 20 different amino acids, each of them is different because their side chain is different. And we can have lots of different types of side chains. We could just have a simple hydrogen atom, or we could have an alkyl group, or a nitrogen, or a sulfur, or a phosphorus atom, and so on and so on. Thankfully, we don't have to remember all of the different structures of the amino acids. And then finally, the central part there, that central carbon, is always attached to a hydrogen, which in this case comes vertically up. We cannot make all of our amino acids. Our body can make most of them. It can make 11 of the 20, but we need to get some of them from our diet. And the ones that we get from our diet, they are known as the essential amino acids. The way that these amino acids are joined together to form proteins is known as a condensation reaction. And in this case here, we have the NH2 of one amino acid on the left. The hydrogen atom from that NH2 group is being removed. And then in the other amino acid, the carboxyl group, the OH, so the hydroxyl group of that chemical, is being removed. And that in a condensation reaction, we're now forming something known as a peptide link, whereby we have a bond between the nitrogen and the carbon atom there. We may hear this referred to as an amide link. Either is acceptable to use if you're asked to either define or to identify a peptide or an amide link. So what I'd like you to do with this one here is, could you please, well, it just says that, draw the protein formed by the condensation polymerization of the following amino acids. In other words, can you stick all those together into one long protein? Okay, so give yourself a few minutes and then we'll go through the answers. So here's a little clue if you're stuck or you want to just check your answers before I show you them. So the hydrogen from the NH2, one hydrogen only, and the OH from the COOH are removed in each instance, and here is the answer. So just pause this and check your work against this, please. So we should have found that we have two peptide links or two amide links. And just a little word of warning, a quite common mistake for pupils to make is to miss out this hydrogen that comes above the carbon that is flanked by the amino and the carboxylic acid group. So hopefully you've got all of that included. It doesn't matter as well, you may see some pictures later on of peptide chains like this, so protein chains, whereby the hydrogen going above the nitrogen there is sometimes pointing downwards. It makes absolutely no difference whatsoever, whether it's up or down. So once we've eaten our protein, so we've eaten maybe a hamburger or whatever, which contains a high quantity of protein, that protein is then broken down, and it's broken down in our body via hydrolysis. This is part of digestion. And the digestive process is catalyzed by enzymes, so digestive enzymes, proteases they're known as. And when that hydrolysis takes place, all of the peptide links are then broken, they're cleaved. And when doing so, we now have individual amino acids. So we've got those building blocks, those monomers are now broken up, are now formed from the protein. The amino acids are sent all the way around the body wherever they are needed, and then they're reconstructed into a different protein. Maybe the burger that you're eating is turned into an enzyme, or part of it is turned into some muscle, or part of it is turned into a hormone, and so on and so on. So notice here the little diagram here that the amino acids are in a slightly different order to show that they are reformed into something different. So this diagram here, this shows the process of hydrolysis. We have our peptide link in the center there at the top, H2O molecules, enzymes catalyze this reaction, and then the H2O breaks apart itself and that reforms or adds on to one hydrogen will go onto the amino group to form an NH2 group, and then the OH will go on to the carboxyl group, or rather it will form a carboxyl group. So we've got an example here of a short part of a protein. Could you please draw the amino acids that will be formed from the hydrolysis of this protein? So pause this and then come back to it when you're ready. So hopefully you found out that there were 3 amino acids here, and these are the structures of the amino acids that were formed. Okay, so time for a few past paper questions. This is the one that we showed at the beginning. Can you circle, or I suppose could you draw or identify a peptide link in the above structure? So hopefully you found these 4 here, any of those 4 there. So the marking instructions tell us that you have to circle any peptide link, which is the CONH, and the minimum accepted structure is the bond between, the diagram there shows something that is unacceptable. So you couldn't just draw the bond, you couldn't highlight the bond between the carbon and the nitrogen. Okay, next question, have a read of this. So what's the name of that process? The name is hydrolysis. Next one. So what are essential amino acids? They are amino acids which humans must acquire through their diet. So B is the correct answer. A bit trickier this one here. So what this question is asking is, can you tell me how many amino acids are being used to make up this part of a protein? To help you out, here is the structure of an amino acid. Okay, pause, and when you're ready, unpause. So there's a couple of ways that you can do this. One of the ways I did it is I tried to find the central carbon, the carbon that is flanked by the amino and the carboxyl group, and I figured that if however many of those carbons I had, that's however many amino acids must be making up. So there's one, there's a second, a third, fourth, fifth, and a sixth. So to me that there are therefore 6 amino acids. Another way you could do it is you could try and see how many side chains you have. So you've got one side chain there, you've got another side chain, you've got this sort of structure there as a side chain, a fourth, fifth, and a sixth. So 6 amino acids make up this structure. So that's part 1 over with. The second part, we're going to be answering this sort of question here. If we have an egg white, well, that's protein. But why does it turn white when it is heated? Or, to give you, in the context of an exam question, it's trying to answer this question here. So to answer this, we need to understand a little about the shapes of proteins. So I showed this picture right at the beginning here, and this is a 3D representation of a protein, and it's really quite a complicated shape, but the way that proteins are built up is in 4 different stages. Our first stage is what's known as the primary structure. Now thankfully we don't have to remember this at all, but I think it's useful to actually go through. So the primary structure is just your chain of amino acids, so the structure that we've seen so far, where you get the amino acids and they all join together by the condensation reaction. Once that long chain of amino acids has been formed, we refer to it as a polypeptide chain. That then forms into our secondary structure, and now the amino acids themselves start to interact with each other, and so the amino acid chain is linked together by hydrogen bonds, and the secondary structure can form one of 2 structures. This is known as an alpha helix, but we can also get something which is known as a beta pleated sheet. Once either the alpha helix or the beta pleated sheet, so that secondary structure, once that's been formed, now what happens is the side chains of the amino acids start to interact. So the purple line there, that represents, let's just say it's an alpha helix here. So our alpha helix here, made up of many amino acids, now the side chains are starting to interact with each other. We have hydrogen bonds being formed between the side chains. We have London dispersion forces. We have an intermolecular force we haven't come across before, a disulfide bridge, and then also an ionic bond. So the structure is getting more and more complicated. We may also get, we will also get a quaternary structure being formed, and in this case now we have multiple chains of the amino acids that are interacting with each other, and this is an example here. This is hemoglobin. The structure or part that we're most interested in though is the tertiary structure, and that's because we have the intermolecular forces which then links it back into higher chemistry. So here's a quick question for you. What will happen to the intermolecular forces between the side chains if we heat that protein? So if we take that egg white and we cook it, what's going to happen to the intermolecular forces there between the side chains? So hopefully we can remember that if you start to heat something, the intermolecular forces are going to break. So they will break, and if they break, the structure of this protein will start to change. And that's really important because if the structure changes, the protein can no longer do its job properly. We refer to it as being a denatured protein. So let's have a look at that in a bit more detail. So here's our tertiary structure, and I've represented our side chains there with Y and X are interacting, and the green dash represents one intermolecular force, and A and B side chains are interacting with a different intermolecular force. Now let's say that we start to heat up this protein. As it's heated up, the intermolecular forces are going to be put under stress, and maybe they will break. And if they break, maybe let's say here that Y and X, the intermolecular force that holds them together, is quite weak. If it's weak, they will break first, and in doing so, the shape will sort of flip out there. It will change it to a different shape. A and B, maybe that intermolecular force is stronger, maybe that's a hydrogen bond, and that therefore is going to keep its shape for a little bit longer, but eventually that might break too. So have a read of this here, please. So this denaturing stops the protein being able to do its job, as I said previously, and it's especially prevalent, and we see it a lot in enzymes, and that if you heat an enzyme past a certain temperature, that enzyme cannot do its job anymore. It can't catalyze the reaction it's supposed to catalyze. So that's pretty much us done for proteins. We're going to go through some questions now, and you can test your understanding of what we covered so far. But to begin with, we'll go through this one here. So the reason why the protein chains unwind, well, we said previously that the intermolecular forces would be broken. The marking instruction says that heat breaks hydrogen bonds, but that was referring to this is a multiple part question, and the other parts show the intermolecular forces. So in our example here, we could have said that just the intermolecular forces between the side chains are broken. Next question. So which of these graphs is going to be obtained? So we have reaction rate, so that's on the y-axis, and the temperature, so the temperature is increasing. So for A, for example, pretty standard graph here for reaction rate. If you increase the temperature, you will increase the rate of the reaction. C, it seems that the rate of reaction has plateaued around about 40 degrees Celsius, but it's still carrying on at quite a high rate. In B, as the temperature increases, the reaction rate decreases until it plateaus. And then in D, the reaction rate increases as the temperature increases, but then at some point it drops off. So which would be applicable to an enzyme? Well, the answer for this one is D. At a certain temperature or above a certain temperature, the enzyme will start to denature. As it starts to denature, it can no longer catalyze the reaction, and the reaction rate would therefore decrease. So D is the correct answer. Okay, so a little bit trickier this one. Take your time with this question. So A is the correct answer, and here's why A is the correct answer. If you look at that section there, and if you imagine that the peptide link in the center, so the first one, if you're looking from the left, if that was then broken, you would have a nitrogen attached to the, sorry, you would have a hydrogen attached to the nitrogen, and it matches this here, and the others, it doesn't fit that at all. Next question. So amino acids are converted into proteins by, the answer is a condensation reaction, so the answer is D. What name is given to the monomers glycine, alanine and cysteine? It is amino acids, and what is meant by hydrolysis? That's splitting a large molecule using water. And that's us done. So we have covered the following. We've given uses and examples of proteins. We've drawn and labelled an amino acid. We've defined an essential amino acid, shown how amino acids can join to form proteins, and then how proteins can be broken down to form amino acids. And we've explained how how proteins become denatured. So if you guys can do the corresponding Google form or the quiz questions, get those back to me as soon as possible. And if you have any questions, get in touch. Bye for now.